It gives me immense sense of satisfaction to host the second annual conference, ACPKCON 2022, Association of Clinical Physiologists of Kerala at Government Medical College, Kollam. We are indeed honored that we got a chance to host the conference. The concept of create, connect, and celebrate was finalized to depict the vast application physiology holds for education as well as research. I welcome all delegates to day one of ACPKCON 2022 to enjoy the academic and culinary feast. So we'll start with session one. Session one is on gut-brain axis, a very relevant topic, and I think today is the most researched topic. Now, we must be thinking, like often we would have come across this phrase, I have a gut feeling or butterflies in my stomach. So why these sensations emanates from the gut? Is the brain and gut connected? Let us hear from Dr. Vikram Gowda, Professor and HOD of Department of Physiology, Pushpagiri Medical College. May I have you on the stage, sir? I also invite Dr. Unnikrishnan, former Dean, Kuhas, currently Professor and HOD, Medical College, Thrissur, to chair the session.
As we go by the words, the Hippocratic, it is said that all disease begins in the gut. All disease, this was said not today or tomorrow, it was said just 2000 years back in the BC, whom we term as the father of modern medicine, suggested that all diseases begin in the gut. Means he had certain evidences to prove that it was, but there was, as a modern day physician or a clinician, I will discard this theory, saying that there is no evidence to prove how the disease is starting from the gut. So many people had doubts and they did not try to follow this. Till long back, even in our own Indian traditional system of Ayurveda and various sayings, we say, we are what we eat. It means what we eat, we are that. It means that there was observation and a feeling that we eat means we are those human beings present in it. Similarly, we have certain unique traditions if you see, specifically to India itself. If you target to the northern part of India, we'll see that anyone going outside for a special event starting in the day will eat curd and a small piece of sugar in it. What is that purpose? What is going to serve that? Or whenever who is preparing for something which has challenges related to memory are provided with almonds. What was the reason why they were projecting or doing that? Something related to memory, they were given almonds present in it. If you come to South India, we have a unique, whatever and how much ever food you have, at the end you should have curd rice. This curd rice, what are the reasons for it? How is that going to help in it? So these were already we were established or we were trying to observe that there is something present in link with our gut and the food we take and something related to the brain or its functioning present in it. So this Hippocrates, when he presented, was not clear. Many were discarded, were not able to follow through it. Not long back, after again around 1000 years later, Galen, anatomist, tried to found that there is a physical connection. When he dissected various pigs and other animal models, he was trying to establish and find out that there is a connection between the brain and the gut. And all these were dissection in the deceased animals, so we could not establish its functionality, but yes, definitely there were some connections present in it, which was established by Galen. And then there was a silent period for almost till 1866, till we see these two pictures which we are seeing on the screen. William Beaumont is father of gastric physiology. Termed as gastric physiology, he is the father of modern gastric physiology is being said. What was he? He was a doctor who was commissioned in Michigan, the northern part of America, and he was an army doctor. And the person you are seeing, that Alex St. Martin, would have never heard it or never known who is this person, but this person was a fur trader what happened to this fur trader was, he was in his just young 20s. He had an accidental gunshot injury. He had an accidental gunshot injury. It was close to the lower part, that is in the epigastrum. And the doctor, William Beaumont, describes that his ribs were broken, his tissue was all damaged in the close region, and his stomach and part of the intestines were damaged to such an extent that he was just able to repair those wounds and clean it, and he felt that he is going to die. So he felt that he is going to die, and he left it. And he was under his observation, but he was just in 20s, but somehow he recovered. He recovered and he started living successfully. But what happened, the problem was, he developed a fistula. And this gastric fistula was such large enough that William Beaumont was very keen and began to observe that certain movements and food particles which he used, initial for 15 to 20 days, were spilled out continuously from the gastric fistula to outside. So he was seeing and food that is being collected outside. But after 20 days, this gradually changed, and he saw that food began to pass into the intestine and passed out. Then clicked an idea, finding for the first time to study the live physiology of digestion. So he observed the changes that meat, when was placed or when he ate meat, that food digestion was faster and going fast forward. At the same time, when he had taken carbohydrate or plant origin food, was retained in the food for a longer time, and digestion process was very small. The experiments, if you go by that way, are very cruel, because in the night every day he used to tie the food to a thread with a small stick. He placed it there, observe for 3 to 4 hours or 5 to 6 hours, then he would take it out and see how it was done. Okay, when you imagine that, you will see very cruel in it, and they are being a form of ethics teacher, I would not encourage, but this was how the experiments were done. So he used to do that. And this was the outcome. So for the first time, not only that he collected the gastric samples, and those gastric samples he sent for the lab and tried to analyze. And this is for the first time he observed and documented that there is hydrochloric acid present in the stomach, and digestion of proteins are helped by presence of this hydrochloric acid present in it. He could not clearly establish how well it is, but yes, the constituents of gastric were well known. Not only these things, he also observed that this person, that is Alexis St. Martin, whenever gets irritated, that change in the food digestion process was also observed. So in his data, he could also document those things, that the change in his behavior also being done in it. And you will say that this was cruel. Yes, this was cruel. In the past days, there were no clinical trials or there were no regulatory bodies to control what was to be done. But understanding physiology and how it is going to happen was not an evidence, but this was used. And since he is termed as father, but there are negative aspects also termed to it, I would like to bring to the notice of the forum also that father of gastric physiology, William Beaumont, got a signed document from Alexis St. Martin that he will work for him for lifelong. Because of which, even though he was doing these cruel experiments, he could not leave this person and go. And almost for 30 years he was associated with him. And after, when he was unbearable for him, he ran away to his home country, that is Canada. But after that also 10 years he did not leave. William Beaumont commissioned him and brought him back, but for a brief period of 3 to 5 years, and then again he left back. At the time of death also, Alexis St. Martin was not peacefully made death. He was then autopsy done for various individuals to find out the reason. I would like to salute Alexis St. Martin, but at the same time, it was the findings of William Beaumont which were been allowed to it. Alexis St. Martin will never be given to this. So these were the first initial steps which were trying to identify that this is how gastric physiology is present, and there is some connection between your brain and there is food which is being processed in it. And this was in 18th centuries, in 1822 to be mentioned very specific. Then come to the era, to the Claude Bernard, where the experiments were done in France, where he had a very big animal experiment house. And this animal experiment house this is the person, Claude Bernard, mainly concentrated how carbohydrate digestion and specifically the role of liver is going to play in digestion. And he also tried to analyze how emulsification of fats is going to happen and feeding behavior of animals. He was a very keen pioneer, and he had rabbits and rats for this model, and he had various animals for it which is being studied in it. So he was trying to establish and trying to study about this metabolism, and as you know already, the milieu interior word being coined by Claude Bernard was because of, after observation of so various parameters, he came to a conclusion that the internal environment should be constant to maintain this. And this was the person who again contributed to it. Just as fun fact about Claude Bernard was, his wife was against him. She led an animal ethics committee association. She would run into the procession all over Europe and ban on all the scientists who are doing animal studies, cruelty on animal studies. Don't ever think only about Maneka Gandhi. There are other people also who had done this far long time back. And later we see that in the Karl Ludwig in Germany, Ivan Pavlov developed the dog's pouch, what is called as your Pavlov's pouch, and he was awarded Nobel Prize for it. He now clearly established beyond doubt that there is connection between brain and your functioning of digestion system. So with these experiments, he was able to prove beyond doubt there is a connection present in it, which is going, it can be both the ways, whether it is directly from the brain coming to the system or something present in the digestive system also informing the brain and getting the feedback, which was established in it. Still, people were not convinced on it. Then in 1980s, when we saw that advent of various radiological abilities came, there were neuroimaging studies, functional MRIs began to establish a clear different model that there is a clear bidirectional talk between the gastrointestinal system and the brain. And to very late recent studies, after 2002, they have been established that disorders in the gut, which are primarily or related only to gut, can also affect or cause certain change in the neuronal connection or talk, which can lead to psychiatric comorbidity. So, in irritable bowel syndrome cases, long-standing cases, when they are studied, they found that there are psychotic comorbidity, mainly associated with depression, associated with it because of the various interregulatory roles played in it. So, with this brief history, we'll try to enter in it and see what and how they are being connected. So to say the key regulators of this gut-brain axis, I'm trying to find here 4 different elements and try to establish my evidence through those. That is, germ-free animals showed that brain is affected in the absence of microbiota, as already sir mentioned, and if you had been keen observer, the PowerPoint presentations also were mentioning something about microbiota. That is, microbes which are being present in our gut system strongly influence. So, in absence of this microbiota, the neuronal change also is observed. That I will try to come in the next slide and establish the fact how it is done. We go to the second component where animals, when they are given with specific strains of bacteria, had alterations in behavior, and human studies of also such strain confirm potential transality of such findings. This can be established to a certain extent in various ethnic regions, geographical locations, and people's behavior related to their food habits. There are certain people whom you say that they are jolly-going persons. We say these people are very arrogant, they are very strong, convinced to it. We say, no, this community or this pool of people are very strong, they are not in it. This was associated or related to is because of their food habits, which confined to have a certain microbiota present in it. And that microbiota is trying to decide what is their behavior present in it. I'm not going to details of it, but just giving a thought process that this can also influence your thinking process and other higher levels of brain functions. Then there are population-based studies. people exposed to infection, most notably, this is one incident which happened in 2002 in Canada, when there was an outbreak. Water got contaminated with Campylobacter and other pathogenic bacteria. When these, normally, when you are contaminated water or you had some other problems, you have GI disorder. You have loose stools for 5 or 6 days, then you follow it up. So these people, when they were cohort followed up for a longer duration of time, it was observed that majority of them turned into irritable bowel syndrome category. There are incidences and reports that after an event of gastrointestinal outbreak due to any of these pathologic organisms, few individuals go to that aspect, that is obtaining or entering into a tract of IBS. But in this group of population, it was seen that it was more number of people. So it tried to establish that when you get this certain bacteria, then there is some common commensals in our bacteria are destroyed, and because of that, the pathological element is causing some abnormal event of our GAT. But in the same cases, when followed up for a longer duration, was found that they had psychiatric comorbidity. They had depression-like symptoms in these cases followed up. And this tried to establish that the more interest, why in 2002 onwards, more interest into gut bacteria was few of these incidents which provoked people to think in a different line, that it is not just the food what we eat, it is the bacteria present in the gut which is causing certain changes or certain behavioral pattern. So in the preclinical studies also we are being studied, or when we in our UG studies we always are said, no, antibiotic is not prescribed here, antibiotic is not prescribed here, antibiotics are going to wash out your gut bacteria, or followed by antibiotics you again have to take lactobacillus to maintain your gut bacteria. So all these are suggesting that your gut bacteria can strongly influence your brain. This is just one slide which I tried to show it out for the first model. As we said, that gut microbiomes have got a very strong influence on brain functioning. So we say that if bacteria are germ-free, there are no bacteria. So how are these animals are tested if you are going to ask or find out? These are mainly in the mouse model. The mouse model, the mother, after C-section, the infants are taken out, and they are maintained in certain environment where there is no contact with any bacteria. So their gut is sterile. They are fed with certain food materials and water, and their surroundings are kept in such a manner that never they are being contact or coming in their lifetime with the bacteria. So how they will repeatedly check is by taking swabs from their oral cavity and from the feces and culture and find what is the bacteria present in it. So if they are contaminated, then they are removed from the trial. So when they observed this, commonly what they observed in this group of animals when they had found is, they saw that these animals lived longer. They lived longer because it was isolated environment. There was no organism present in it. Definitely they lived longer. A different component which they found very keenly in observing in these animals was, they found their immunity was very weak. When there was a control arm and testing arm and they were infected, they found that their immunity is very weak in it. Their various parameters like blood-brain barrier was deficient. The blood-brain barrier was not well established, because of which they had deficient in the blood-brain barrier. There are multiple synapses and dendritic densities which are being formed in various neurons. Not all the places. There are different relations. There are one side as well as the other side. You will find different associations present in it where were deficient in it. So all these entities inflamed or tried to establish that the gut biome had a strong influence on the human brain, whether it is proliferation or enhancing or establishing reflexes or establishing inhibitory neurotransmitters present in it, and immune related specifically to the brain, which has been studied in it. Since it is gut-brain axis, I am trying to concentrate only on to the brain and their effects which are being present in it. They were even establishing their memory and their strong reflexes being studied in it. If you just go by the amount of bacteria being present, So in prenatal life, there are studies which have been trying to establish that at the time when the placenta is formed itself, there are certain bacteria which present in it. They are contrasting, not so well established, but still there was some amount of bacteria present in it, definitely after the birth. And if you see the study done, we'll find that people who are born in cesarean section, born with vaginal delivery, will have a different entities because they are exposed to different types of microorganisms when they are born through vaginal delivery. So you will find that the aspect of gut microbacterium and bacteria which have been present are different from what is being present in the occasional. As you go throughout the lifespan, in the adulthood, it is the maximum amount of the bacteria which are present. If you just go by the number, if you find it is almost million times of what is being present in it. And if you just go by the genetic combination, if you have presence of the genome present in it, we have 99% more, that is million number of times more genes than that of our own genes. So those many number of bacteria are present in our gut. So they are going to influence by various ways, whether it is neuronal proliferation or dendritic arborization, neural mitigation, which have been studied. But all throughout the life, they are having a significant effect on synaptogenesis, synaptic formation, as mentioned or seen in the slide. I'm going to stop here with the bacterial intervention because tomorrow's topic is again on that microbiome. I will not be talking much on the bacteria related to the gut, since physiologists will try to establish what is that physical connection or chemical connection which are being present to it. My understanding and reading for a long time when we are seeing about as a GIT, if I see, even though GIT is present or arranged in our body and present intrinsic, if you see or you try to analyze as perspective with the body, it is always outside because there is a lot of water is being secreted for digestion and absorption. At that time, all this water is released or being evacuated from extracellular fluid volume. So if it is lost, it is lost. If it is present in GIT, it is not present in your body. It is present outside. Whatever is the nutrition, even if you take poison and you don't have receptors present in it, this poison is never absorbed. You are still safe within your body. Even though it is present, it is said that you are present in your body, it is never absorbed in your body. So, similarly, if a toxin is absorbed, then it enters into the circulation causes. So, for all practical aspect, this long track of GIT, even though we say it is being situated inside in it, this practically for our body is communication from outside. It is talking from outside what is being happening in it. So, we will try to study with that aspect how is that talking going to happen and how it is related to it. So these are the important aspects already being highlighted and told that communications is happening to GIT to the outside. That is from GIT to your body. That is the outside of GIT. So outside of GIT is happening mainly by autonomic nervous system, enteric nervous system, immune system and neuroimmune mechanisms. And there are neurotransmitters directly related few playing in it. Branch chain amino acids, bile moieties, short chain fatty acids, hypothalamo-pituitary adrenal axis. Now my talk will not go in details of it. I'll take only each one of it and briefly try to establish how these are linked or connected to each other, and that will try to introduce our topic for these sessions. We'll say autonomic nervous system. What comes to our autonomic nervous system when we strike autonomic nervous system? All those organs which are not under voluntary control are controlled by autonomic nervous system. So what is this autonomic nervous system? Autonomic nervous system primarily, if I say, it has got a sympathetic branch and your parasympathetic branch or arms. What is this sympathetic and parasympathetic? All organs which are controlled by our autonomic nervous system have got a basal functioning aspect. They will do their functional aspect on a basal mode. You want to increase their efficiency, you will find by one of these arm regulating it. You want to decrease the efficiency of that arm and another arm of it. So these are your two reins which are tied to your horse. You leave to your horse, it will be running, so your organ is also functioning. You want to go in a particular direction in a very high speed, then you stimulate it with one of the arm, either your sympathetic or parasympathetic, based on its input on that organ, and you want to control it, run in that direction very gradually, and you put the other arm into the action which controls in it. If you go by the sympathetic and parasympathetic, I'm not again talking into the details of it, we'll try to establish that gut as parasympathetic is vagus nerve. You leave to vagus nerve, there is nothing else present for your gut. Because parasympathetic outflow, if you just see by the parasympathetic outflow, 70% of the parasympathetic outflow is through vagus nerve, and the vagus nerve is the strong and it is innervating all the parts of the gut to almost to the transverse colon, 2/3 of the transverse colon from your oral cavity to your colon. At exception of certain facts, you will find vagus nerve is the one which is playing role into it. So we just talk to the aspect of the vagus nerve. Now we see that how is this vagus nerve going to? As we have established or tried to establish by various animal studies and models, we have suggested that vagus nerve has got sensory input information received from the gut, whether it is in terms of chemical nature of the substance you have taken, or volume which is being taken, or osmolality of that substance you have taken. All these parameters are judged. So there is sensory input coming to it. Similarly, vagus has also got two-way communication. So it causes increase in the secretion of substances which will help in digestion of it, which has been established as we see cephalic phase or pre-cephalic phase, cephalic and gastric phase. We'll find how we are going to affect these stimulation factors present in it. This slide which I have presented is mainly suggesting vagus nerve is not just, it has got a 3-level of area where it has got direct communication with the muscle fibers, it has got direct communication with the interneurons present within these muscle fibers, whether it is submucosal or with the enteric nervous system. Apart from that, vagus is unique, it has got direct connection with the mucosal cells also. So, to an extent, vagus is so much deeply connected that it can regulate its function either by of this or by all of this. So, it can directly go to the mucosa and cause certain change, or through your enteric nervous system it can cause change, or by influencing enteric nervous system outside it can cause change. So, this pathway which I have tried to highlight or show is your cholinergic anti-inflammatory effects of vagus. Now, cholinergic anti-inflammatory effects of vagus are the recent studied models which you are being seeing, that inflammatory properties which are generated in the GIT or other aspects, all those bowel inflammatory disorders, vagus plays a predominant role in decreasing that anti-inflammatory effect. And how is that is this effect which has been tried to show. If you see that there are connections and some dotted lines present, these dotted lines are mentioned that the clear evidence of established is not being maintained. What are they going to do? They are going to control the cytokine release, specifically in the GIT, and at the same time release of stressor hormones, whether it is norepinephrine from the adrenal gland, as well as their metabolism and their action in the spleen. What is their action in the spleen? In the lymphoid is to alter your lymphoid organs, specifically dendrite cells and their expression, and causing on the plasma cells to release certain factors. Again, interleukins and other intermediaries of the inflammatory has been regulated. So in this one nutshell word I want to try to mention is vagus is not just a nerve which is responsible for carrying your information and delivering certain information to the GIT, but also plays a role in healing wherever there are problems associated with GIT by systemic intervention. If you go to the system, sympathetic nerve, if you see as per the innervation, this diagram tried to more of the innervation present to it. Go by the innervation, we will see that predominant innervation of sympathetic comes to blood vessels. And it is a strong controller of blood flow to a particular organ. Apart from that, sympathetic, if I try to see, it is predominantly or strongly on to the sphincters. So these are the two areas where your sympathetic nervous system is going to intervene and do the action. Not like vagus point to point, every part of the organ, as I mentioned, it is in the mucosa, or it is in the submucosal plexus, or it is in the intrinsic ENS, there is innervation. But sympathetic is predominantly failing or trying to maintain or cope up with that. And we'll find that with the relation to the blood flow, it alters the regulation, and that is how it gets feedback. Again, in sympathetic also, you have got two-way communications. It receives the information and responds to act to it, mainly on the volume based related to it, and the actions presented are related to movement and the blood flow. The enteric nervous system, the second aspect, which we are saying that this is one another important aspect which is communicating to it. In enteric nervous system, if you can see, there are layers, whether it is submucosal layer or lamina propria present followed in it. We have submucosal plexus followed by your muscular layers. In between the muscular layer, you have group of neurons present in it. What are your Auerbach's plexus and Meissner's plexus, which we commonly term them. So these are the layers present in it. Enteric nervous system itself is called as mini brain, because the amount of neurons that are being present in enteric nervous system is almost as huge as that it can function on its own. As previously introduced, that how our GIT is going to function on its own, if you take the innervation off, even your sympathetic and parasympathetic is taken off, GIT can absolutely perform very well. That is why this direction is also said, oral anal direction is also mentioned in the chart for the reason to specify that the cells which are present in abdirection and oral direction respond in a different way to a particular hormones or neurotransmitters or their actions present in it. So this is how this is going to intervene and present in it. So enteric nervous system in itself can function and perform as an excellent individual present in it, and this also influences the microbiota present in it or the stimulus which are being present role play going to present in. There are various intervening neurotransmitters and the hormones which are being stimulated and present released through it. In this slide, I tried to establish a fact that what we studied, the autonomic nervous system and the enteric nervous system, are not separate enough, they are interlinked. You say whether it is sympathetic nervous system or parasympathetic nervous system, they fall on synapses with your enteric nervous system, and that influences the outcome. So whenever you cut off the signal, that is when you cut off the signal from the autonomic nervous system to the enteric nervous system, they may slightly be unresponsive, but after a period of dormant state, they start functioning again. When they start functioning again, means they can establish and maintain their own. So they have their unique features, whether it is respect to volume or osmolality or chemical substance, they can function and perform to an excellent state. The third most important part, which is forming an axis with the brain, is your mucosal associated lymphoid tissue. This mucosal associated lymphoid tissue, if I go by the histology, then we'll find there are Peyer's patches and there are group of lymphoid follicles present in it. Go to the details, you will find more and more area. There are subfollicular region, there are follicle associated epitheliums, there is M cell, antigens and all others. Don't go into the intrinsics of it. I'll just try to tell in brief in 2 minutes what is exactly in it. If I go just by the number of plasma cells, 90% of those plasma cells are present in our gut. It means what? The exposure of or development of these immunoglobulins to produce against an antigen is primarily learned in our GIT. It means what? The GIT is the place where interaction happens outside. Yes, definitely on the skin there are present, and the studying of this skin microorganisms and this development of immunity is far, far less when compared to our GIT. GIT has got so much, that is 90% of your plasma cells are present here. It means what? What is that indicating? It is just indicating how large. And this is trying to process, means whatever is the antigen, whether it is in form of food or it is in form of microorganisms or in form of various others bacteria. If you see microorganisms, this gut is not just microorganisms. You can host protozoas, you can host elements present in it, you can host various numbers present in it without any much problem, unless they begin to affect your physiological aspect and derail it. You have various aspects present, all this present in it. So this immune system is studying. So immune system, when they are studied and they are formed, are they going to remain there itself? No. This immune system now began to release interleukins and various other mediatory factors which will affect your general immunity. And that is the reason what we tried to establish in the earlier mouse models, that the immune which were less is because of this interaction. It can be contributed because of the bacteria also and the other food substances which we are taking into it. If you go to a certain extent, our body responds reflexly to it, whether it is associated with immune or not, or directly the neuron also can be present. Now, for example, if I take taste receptors, we say taste receptors are present only in the oral cavity. If they are present in the oral cavity, your taste would have been hurt. But if you are very hungry, you don't have any other food, but the food is rotten, you eat that food. You don't appreciate the taste because you want to fill your stomach. But after 5 minutes, you vomit it out. Why did you vomit it out? Who decided that this food should come out? It is because we have established that there are taste receptors even in our gut. Not only in stomach, up to the intestine, they have been determined to have taste receptors. And these taste receptors there itself, that is why now you give the reason that a drug which has got an irritate or which is not edible causes vomiting even after 15-20 minutes or half an hour of it. So this taste receptors causes this feeling which causes that food to be thrown on out. So this immunoprotective effect of the gut is related both to your reflexes as well as study of these food particle chemical substances present in it. And this is just in relation to the microbiota, because we studied is only with the food which is present. Where there is no microorganism which is studied, it can be acting in it. But the same adaptive, whether it is innate immunity as well as adaptive immunity, both innate and adaptive immunity can be greatly influenced, and they can be able to develop and communicate to our immune system because of the gut microbiota which are releasing various substances. And there are neurotransmitters, the fourth aspect present to it, which are playing a role. The list of neurotransmitters goes endless, but trying to find out or restrict to few of them which plays very important role. Serotonin comes first among them because almost 80 to 90% of serotonin which is in our body is again accumulated or being formed in our GIT because of the various actions. And that will be helped even by our microbiota. And as we go to study this microbiota which is releasing the serotonin are entering into the circulation, and that will have effect in the brain also being studied. And various psychiatric illnesses which are being studied are also related to this serotonin release from our GIT. Acetylcholine, a primary neurotransmitter associated or equipped with the vagus, plays a role. But apart from that, there are interneurons present within our GIT which plays a very keen role and signals the change. And we saw, as we know, acetylcholine receptors related to muscarinic or nicotinic are present. Specifically to autonomic nervous system, we say muscarinic receptors are abundant, and in sympathetic also they can influence and cause change to present in it. Norepinephrine is the predominant one which has got the effect, because whenever there is stress, there is lot of release of neurotransmitters, and this norepinephrine, the effect which has been caused in it, I'll try to highlight in the last aspect where it is that is hypothalamo-pituitary axis. I'll try to highlight and mention how it is. But norepinephrine itself has also got an effect role to play in it. It will affect the signaling to the outside, that is organs and different gamma-aminobutyric acid, another strong inhibitory neurotransmitter which plays in neuronal and all the spinal communications will find gamma, and this is also abundantly produced in our GIT which has got effects on systemic effects. This is just one slide to overcome that, where they are produced and how they are produced, but this I have also already mentioned that they are abundantly produced in GIT because of the interaction with the microorganisms present in it, and they can influence these changes. gamma-aminobutyric acid, mainly released in the central nervous system, but they have the effects in GAT also because of the interplay present in it. Norepinephrine, main spinal cord and sympathetic nerves, which is being present in it, but they will affect your movement of GAT and intestine present in it. They are two-way communication. They are affected from here also, from there. Acetylcholine, as I mentioned, system, CNS, autonomic and others, will also affect systemic organs and their functioning ability, and serotonin. Coming to the smaller aspects present in it. What are these branch chain amino acids? These branch chain amino acids are essential nutrients, mainly leucine, isoleucine and valine. Now what are these? These are essential amino acids which cannot be synthesized in our body, and they are a very form an important component. Where are they derived from? They are derived from meat, dairy and legumes. What is their role? They will influence mainly the functioning of GAT, but the theory of consuming this branch chain amino acid was very rampant, and bodybuilding and nutrition people began to start consuming this branch chain amino acid to a greater extent. But it has got its own toxicity effect, and they are abundant. Too much of anything is also not too good. So this is branch chain amino acids playing the role in it. Bile moieties. Diagram is confusing. Don't follow to influence in it. But what I have tried to establish or mention in this diagram is what will happen to your bile salts, how they are formed. So when you see cholesterol, fat which have been formed in your diagram, they are been broken down. Now they are broken down to ultimate formation of taurocolic acid or deoxycholic acid and cholic acid. These also are being influenced or being modified by the presence of bacteria in it. So what happens if this bacteria is not present? What happens to that cholesterol if it is being left out in it? There is an alternate pathway, a neuronal cholesterol clearance pathway, which can affect the functioning of various neurons. And recent studies are trying to establish that related to Alzheimer's and related to multiple sclerosis. This bilirubin pathways which are being done or influenced by it has got certain effects related to the bile moieties present or contributing to it. Short chain fatty acids. What are these short chain fatty acids? Short chain fatty acids are purely carbohydrates, and only carbohydrates which are undigested food carbohydrates. These undigested food carbohydrates are broken down by our bacteria, and primarily they are going to form acetate, propionate and butyrate. Now, what is the fate of these three which are being formed? All these three can be put back into circulation. So acetate can go back into circulation. It provides us an energy pool. Propionate and butyrate also can be put into the circulation present in it. What other things they are going to affect is your signaling pathways related to fat metabolism and maintenance of that gut bacteria, and prevent other bacteria to growth also will be helped by many of these short chain fatty acids present. Short chain fatty acids are influenced to associate with oxygens and non-oxygens, because primarily our GAT was concerned was for what purpose? The neuronal innervation to our GAT was meant to just whether to eat food or stop eat food. That was the main concept. Whether you have to feed or satiety, I am full. Is that all which has to be done? No, it is beyond that. That is what is being tried to establish by these connections present in it. The last, which is recently and the hot debate topic which is going on, is hypothalamo-pituitary axis. If you go as the physiology aspect, we'll see that hypothalamus, which is non-neuronal mediated, humoroendocrine related, we'll find there is release. Whenever there is stress, what is that going to cause? Any stress related, whether it is food or it is neural or it is any other type of stress, what is that going to cause? That is going to stimulate your ACTH. And that ACTH releases your cortisols, which has got tremendous release on the effects. What is this cortisol going to do on various effects, whether it is on the GAT or in the peripheries? It is going to produce more amount of epinephrine, norepinephrine. This epinephrine, norepinephrine itself, when it is left into circulation, causes more damage. But at the same time, presence of microbiota in our bacteria helps in releasing or reducing the amount of norepinephrine put into circulation by alternate mechanisms, because of the various mentioned above, whether it is short chain fatty acids or your branch chain amino acids are playing a role related to it. And coming to the aspect, this I am trying to highlight and show that no individual aspect is alone. I cannot explain any physiological aspect as only by that, and that is the one which is going to cause change. If I am saying that hypothalamus and pituitary causing CRH and release of this cortisol and all cortisol, they are got its effect, but also they will be stimulating microbiota and cause certain changes, which in turn will alter your immunity, which not only alters but also will alter your neurotransmitter release. And these neurotransmitter release which are present will also alter your autonomic nervous system. It will alter your enteric nervous system, which in turn will influences the outcome of that body. Coming to the last slide of this gut-brain axis, what does that? This is a self-explanatory slide, which you are seeing that it is two-way communication. It is not just one way. You will find from the mucosal cells to the enterochromaffin cells or to the lymphoid cells, you will find there is communication. There is communication between these layers also present in it, and there are communication present with the gut, which are whatever, whether it is food or microorganisms present in it, and this influences strongly your brain. And what related to your brain, whether it is appetite or energy homeostasis. That is our primary concern. Our gut is present not because for other functions, that is for our food, serving the purpose, brain and death. But apart from this, what other factors which are affected are our emotionality and mood, resilience, stress resilience and recovery, learning and memory, and sensitization and pain. Even though I have not talked much about the spinal cord has also got equal and strong important role in this system, because when we see as per the name, the pathway of pain and traveling will take place predominantly from the spinal because it is appreciated. Whereas the sensation of volume or the sensation of osmolality is never appreciated because it is predominantly by the vagus. So it's a dual component, and that is why it is always remains in medical field the abdomen Pandora's box, because you find something, feeling is something, and when you enter in, you find something else present inside than it. So with this, I conclude my session. Thank you.
Now I request Dr. Unnikrishnan sir to hand over the memento to Dr. Vikram sir. Thank you, sir. Now I request Dr. Premlata madam. I request ma'am to hand over memento to Dr. Unnikrishnan sir.